Good morning, and we are on for this morning's lecture. I will turn the webcam on. I don't know how useful that really is, but here we go. I really need some kind of a diffuser for that. Anyway, today we are going to continue through the rest of chapter 12, and that's it. I will have the take-home midterm available by this afternoon or early this evening. Not long after I complete this lecture and I upload the Bio 101 midterm grades. I'll look at throwing that together. So it might be after lunch with the boys, which I don't remind them of it, but they keep asking me, so I guess I'll keep going because eventually they're going to not want me to be at their school having lunch with them. So I'll take what I can get now. But anyway, we are looking at the rest of chapter 12, starting with section 3. Sorry, like I was finishing saying, I'll have that uploaded as a document later on, and you can email me. You can write it on, like a document, a PDF, preferably, or more easily. You can do it on paper, and then you can use, you can convert that to PDF. There's a fun Adobe Scan app for Android anyway that will turn your documents into, your pictures. It'll scan them and assemble them in a PDF, but that's a little complicated. So anyway, typing it up is probably the easiest way, right? If you really want to do it on paper and put it into my inbox by Friday, that's fine too. Because why not? I haven't gotten mail in a while. All right, so here's what we're looking at, where we left off on Friday. And this is looking at whole genome methods, and then some suggestions of pharmaceutical applications for these whole genome methods. Now, there was an X Prize that was worth $10 million. I don't think it's not going to bother me. I'm sorry, $10 million, an X Prize for the development of a process that would be able to process 100 genomes rapidly. I'm just going to unplug that because that is kind of annoying me. No. How do I work this? Where is my large automobile? I've got it running in the background, don't I? I didn't get the sleep I thought I did. Okay. No steam. All right. Make sure I'm still doing what I think I'm doing here. Am I still? Yeah, I am. Okay. So there was an X Prize. It was canceled for doing whole genomes cheaply, and it was canceled because innovation outpaced their X Prize. So they didn't feel like there was a real inducement to innovate. As it is now, you can get your whole genome sequenced for under $5,000, and it will turn around in just a few days, maybe a week or so total, to do the full assembly. It's a technology that has allowed us to gather a lot more data than we've been able to process or figure out how to, study that, because it's kind of the cart before the horse when it comes to experimental studies. You have to do observational studies on these large data sets. Anyway, okay, so whole genome methods and pharmaceutical applications of genetic engineering. So the uses of genome-wide comparative analyses, and then the advantages of genetically engineered pharmaceutical products. Okay, so basic terminology here. We have our, when we say genomics, we're talking about the study and comparison of entire genomes. And when we talk about transcriptomics, that's where we look at the entire collection of messenger RNA produced by cells. It's also used to monitor the gene expression of virulence factors. Metagenomics, metatranscriptomics. So when we apply the study of genomes and collections of messenger RNA to entire communities, that's the meta aspect. And then there's the field that's still reasonably new, right? Pharmacogenomics and toxicogenomics. This is where we're able to evaluate the potential efficacy of a drug or a toxin based on an individual's genome sequence. Well, so that sounded kind of weird. So in toxicogenomics, you'd be looking at particular vulnerabilities in an individual. Hopefully that's not a bioweapons field. So proteomics. So we look at the entire complement of proteins, or the proteome. And Dr. Ricky Cox is pretty big on that, if I remember correctly. So one of the common reporter genes we use in molecular biology is the green fluorescing protein, originally isolated from a jellyfish. Here's an example of its application in mice. And so here we've done some kind of a genetic modification where we've added the GFP gene to some protein that commonly expresses, obviously, in the skin. And here's 2 mice that are expressing that and 1 that does not. So as a reporter gene in bacteria, it works well too. And then if you remember the molecular cloning bit before, that's a terrible transformation plate. I don't know. That's awful. I'm going to have to go out of frame for a sec. I mean, there's some white colonies there you could pick off. There's these big white colonies here. And maybe those are your transformers, but look at all the little tiny white colonies. That's an overgrown plate. That's what I think it is. And I know that because I've seen overgrown plates. You see some of the dye here from the X-Gal. It's kind of leaking out into the Aaargh. Got these little halo things going on. Yeah, so those big colonies, you have to take that and streak it on another X-Gal amp plate to make sure you weren't picking up anything else, because those are big colonies with very close neighbors. And then from those single isolated colonies, if you still had no color, then maybe that has your transformant. Maybe that's carrying your vector that's carrying your insert. Another technology is RNA Interference Technology, which is quite interesting because with so here's a cartoon for the eukaryotic cell, right? So and this is true, they commonly make small antisense RNA or interference RNA, RNAi, and these are complementary. So these are transcribed from the non-template strand. So it's the strand that looks just like the final messenger. No, not the final messenger, but the pre-messenger at least. So the antisense RNA, when it's bound to an RNA molecule, you have a couple of effects. For one, you can inhibit it. It can't be involved in protein synthesis anymore directly. But for another, it can also be used to extend the lifespan of that particular messenger RNA. That's RNA interference. So you can do these studies where you design or you use an interference RNA construct of some kind, where you can silence genes without altering the gene in the genome, so that you can show a demonstrable phenotype change based on gene silencing, where you can alleviate that silencing and then thus restoring the original phenotype. So dicer and slicer and all that you're going to learn about in one of your molecular biology classes. But we're looking at microRNA and silencing RNA here. Here's an example. So we have the microRNA that is binding to the messenger RNA, and it's able to block transcription. Sometimes microRNAs are used to increase translation rates. And then silence, so this is an imperfect match, and then silencing RNA is a perfect match. this enzyme is making this happen here, and that's leading to cleavage with dicer and slicer, dicer. But anyway, the point of this is to demonstrate microRNA and silencing RNA. The rest of that you'll go through in a future course. Okay, that's the basics there. And now we're looking at gene therapy, in which we are making progress. Okay, so the mechanisms, risk, potential benefits of gene therapy, the ethical issues involved, and somatic cell and germline therapy. So first, one benefit of all this sequencing technology is that we have the ability to do genetic diagnoses. And one example is with breast cancer, the BRCA alleles that dramatically increase the risk of having breast cancer. And knowing whether or not you have the alleles that put you at risk, some people make the choice to be proactive and minimize their own risks. The California Department of Health, and I'm sure California is not the only state that does this. This was the one that I grabbed, has a newborn screening program where they'll screen for, among other things, we'll see metabolic disorders, endocrine disorders, hemoglobin disorders, various genetic disorders. It's a state level program. And at this point, the results of this, and some of you might want to be genetic counselors at some point, but the results of this is when you find out you have this risk, or you find out a newborn has a full-on condition, the discussion with the parents or caregivers then turns to management. We don't have the technology to cure these things just yet, though we continue to work towards that. But basically managing expectations, therapies, adaptations, things that you can do. Now in California, this screening is for 80 different diseases. Gene therapy, where we're at. So we use a vector, because remember, transformation is facilitating the uptake of naked DNA, even in bacteria, takes a lot of effort. Cells aren't tending to want to do that on their own, but viruses already know how to do that. So the adenovirus is a popular vector for these attempts. And adenovirus in that group, they can cause the common cold along with rhinoviruses, sore throat, bronchitis, pneumonia. It can cause viral conjunctivitis. Oh man, that's right. We're going to go over that after spring break. That's fun stuff. Induced fever, bladder inflammations, neurological disease. These viruses are capable of doing all this in part because they're able to bind so readily to eukaryotic host cells. And that's an interesting picture because they get... Oh, okay. That's the virus binding to the nuclear membrane. No, that makes sense. And binding at a nuclear pore specifically. Interesting. So with the genetically modified adenovirus vectors, we can put our target DNA flanked by the viral DNA that will help this... that'll help target it into the nucleus. This is what you're getting out of this viral DNA. You don't want it to do all the other virus things. You want it to just deliver its package and then get eaten. So this one large study that I took all this from, I only had data up to 2014, but and there was a dip in gene therapy clinical trials approved in 2014, but I don't know where that's at right now. I know it's in the hundred, I think single hundreds, as in below 200 right now, that have been approved in recent times. And I think a lot of these are probably happening outside the US because we apparently don't want to do science here anymore. So anyway, the point here is that it's overall a growing trend in the number of gene therapy attempts. But this goes back to 1989, so we barely had PCR back then. but attempts were being made. So the things we're trying to address using gene therapy, cancer is number one, as you can expect. And as an aside, well, I don't know if, yeah, I might forget it later on. I interviewed at U of L for med school as after not being selected for the MD PhD program. That was my fallback, right? I wanted the PhD first. I know that's backwards, but that was me. And I can suggest one of the number one thing you should probably know when you go into that interview, probably any med school interview, is be up on the current stats of disease conditions affecting the population, particularly in the state where you're going to med school. And for Kentucky, of course, it was obesity and smoking. And that probably hasn't changed, but, you know, be up on that when you go in there. I was not, but that one should have been common sense, right? Anyway, so monogenic diseases. So these are diseases that are caused by a single mutated allele. Makes up the next chunk, but not, which isn't near as big as the cancer, of course. And then infectious disease, cardiovascular, neurological, ocular, inflammatory diseases. That'd be nice if we had some good treatments for some of those autoimmune disorders. But again, by and large, cancer is what we're going after. So the phases. Now remember, wait, is this something you're remembering, or have I even told you yet? Well, I guess I'll review it real quickly right now. Phase 1, a phase 1 clinical trial, is where you're using a small group of participants, and the primary goal of the study is to evaluate the safety of the therapy, that you're not making anything worse. You're not harming people. You're not even looking at evaluating the efficacy of the treatment, though you're certainly collecting that data. The main point here is that, is the treatment safe? Or is the risk manageable within, you know, safe parameters? When you move into phase 2, after you've established that, then now you're looking at the efficacy. And again, you're still using a small group. When you get to, and if you pass that, then you get into phase 3 clinical study. This is where you're going to try to do your randomized treatment groups, you know, the double-blind study approach, and you're going to use a lot more people. So you're still looking at efficacy and safety in a wider population. And then by the time you get to phase 4, this is where you enter what's called post-marketing surveillance trials. This is where you release this in some way to some portion of the public at large, and you start collecting data on how it goes. And we do that. That's sort of permanent phase 4, right? We always monitor and do surveillance on the efficacy of any kind of treatments we've approved. Okay, so, but as you can see from here, a few years ago at least, the vast majority are in phase 1. So they're just doing an evaluation for safety. And then there was only a couple in this meta review, or this just a straight review article, I guess, that had actually made it to phase 4. And a few were actually single subject. So that's interesting. So vectors used in gene therapy clinical trials, adenovirus, which I mentioned before, is the most popular. Retroviruses as a group is also right up there with adenovirus. And while I said transformation is difficult, and it is, it doesn't stop people from trying to use it. And then we have other virus vectors that people are trying to use, including the pox virus, the herpes virus, and some other vectors. Yeah, so viral vectors seem to be the best way to go. Remember that CRISPR-Cas9, really cool, but if you have a functional P53, it's not going to work very well on you. CRISPR-Cas9 will not. Now, here is where we get into fun DIY biohacker land. I don't know if I mentioned this one already, but this guy documented his own gene therapy. What he was trying to do is use a viral vector to cause make his lactase gene become persistent. So to eliminate his own lactose intolerance. to turn himself into a lactose persister. And he says it worked. So that's interesting. This is Justin Adkin. And God, there was something else he was trying when things started going over the edge with his Ascendance Biomedical company. And I'd be speaking, I don't know. I don't remember exactly, so I'm not going to say. But it's not good. That company went under, though, and with lots of drama around it. Now, this guy, Josiah Zahner, he has a PhD, but he was selling kits using CRISPR-Cas9 to try to inhibit the myostatin gene, and I think I already told y'all about that, so I won't belabor that point. And so he's claiming that you could do this on your own in your garage, and you can, but in my personal opinion, the vast majority of people are only risking infections, right? Realistically, if you start injecting yourself with this construct that you think you've made. But now we know that it won't even work particularly well, so that risk is completely unwarranted. I mean, that doesn't keep people from trying these things, but I recommend against it. Like, if you hear anyone talk about doing their home brewing, and they talk about how it's really difficult and they have had lots of problems or whatever, the vast majority of the time, those are going to be people that don't understand aseptic technique, like y'all do by now, hopefully. You know, really it's as basic as keeping things clean, and that would be true if you were trying to... Wait, I'm not trying to talk you into that at all. Sorry. Anyway, you probably shouldn't run around injecting yourself with anything unless you've been trained to do so, like if you're insulin dependent or whatnot, or you have other medications that you've been taught to self-deliver. You definitely shouldn't be making up transgenic constructs in your lab garage. Though I'll tell you, if you don't know about Parafilm yet, you're gonna love it. Parafilm is awesome. He has all the right props in the background for this stuff. But anyway, now I already mentioned transgenic fish, so I'm not going to go over that again. But let's talk about transgenic goats for a moment. Here's some applications of this biotechnology. So I don't know if you remember hearing about this 10 years ago or so. The idea was that we were going to make super silk steel material, and that goats were going to produce it. There were some jokes about the spider goats. Of course, now, thanks to Facebook, there's people that think that was a real, like, spider goat. And I thought people saying that everybody understood that was a joke. I don't know, man. Apparently not. Anyway, these were goats that have been engineered to produce this dragline 1 protein. The gene came originally from the golden orb weaver, and they would engineer this protein in their milk. Now, the milk wouldn't come out stringy or anything. It was just free-floating individual protein units. There was an extraction process that would actually get it to polymerize. But this was supposed to be a biosteel polymer, but it didn't work. And there are 30 transgenic goats left, and they're in a breeding program at Utah State University. If you're interested, you'd have to go to Utah, so I don't know. Yeah. I've been there a few times, but I don't know. Personally, I can't recommend it. But the person that's overseeing this is Randy Lewis, if you're interested in working with transgenic goats. Transgenic plants. This is a fun subject. So there's the generic blurb on this. So corn, we have a bunch of modified corn, often modified through plant biotechnology. But I'm going to go into a little bit more detail, sort of. So here are the things out there that are genetically modified, that are approved for growth and consumption in the U.S. We have the apple, which I'm going to mention. This has been, it's got a non-browning. It won't brown in the air. So, like when my kids used to get Happy Meals, now they're all into wanting double quarter-pounder cheeseburgers, and they're like 9 and 11. My 15-year-old still loves nuggets, so he hasn't really changed. But the other two, they want the adult meals now, and they usually eat it, so, fine. But anyway, when they used to get the Happy Meals, they would always choose apples. So I'd get a moment of, ho, we're fancy because our kids choose apples out of that. It wasn't encouraged or discouraged or anything, whatever. But anyway, there had to be something on those apple slices to prevent them from browning in the air. Because you know that your average apple, you cut it up within hours, you're going to get some kind of a browning, at least tinged with brown. So now we have this genetically modified apple that won't, because the enzyme that facilitated that, that caused that to happen, has been edited out. It's been knocked out, so it's not there. Potatoes, we have a bunch of different traits. Reduced bruising and black spots, low acrylamide, blight resistance. So, yeah, these are things that we have out there. So field corn, field corn has a lot of other uses that aren't necessarily directly eating the corn. We have a lot of cultivars out there that have various types of insect resistance, herbicide tolerance, drought tolerance, and they're used in all kinds of industry. I'm not a fan of ethanol as a fuel, and I don't have that in here. That's a different presentation, but I think if we were gonna, I like the biodiesel approach with the modified algae. I like the way that's shaping up quite well. I like the syngas, the synthetic gasoline approach, which isn't based on genetically modified anything. But also I like the butanol. We used to use butanol a lot to make acetone, but another product that came from that process, from the Clostridium acetobutylicum, was butanol. Now butanol stinks a lot, but you can pour it directly in any regular gas tank and it'll burn almost as efficiently as gas. Now we moved on. We don't make acetone by the same process anymore. We don't ferment it, basically. We don't use bacteria to produce it. But we have that technology. That was in the 1940s. We were doing that. All right. So anyway, field corn. It does get involved in processed foods and cereals and whatnot. And of course, making alcohol, which is ethanol. But that's fuel ethanol. Which, I don't know, it's kind of silly. When we make the ethanol from the ethanol plant, from the corn feedstock or whatever, we have to dope it with gasoline to call it non-drinkable. That's what separates this from calling it just alcohol. And we have other uses for corn. Yeah. So we have canola, herbicide tolerance, and this is glyphosate is by far the most common type of herbicide tolerance in any of these. That's the Roundup Ready trait. And that is the first generation of those have come off patent. So now anybody, you can grow the first round of Roundup resistant cultivars. You can just buy the seed and do whatever you want with it because it's no longer covered under the patent. The first version, version 1. So we also have alfalfa, herbicide tolerance. This is probably also glyphosate. Soybeans, so all the herbicide tolerance here is going to all pretty much be glyphosate resistance. So we have some insect resistance, and insect resistance in a lot of these is the BT, the Bacillus thuringiensis, which is able to make a toxin that is really bad for an insect stomach. It's fine for us, but it's also used extensively in organic farming, except as a live bacterial culture that gets sprayed on 2 or 3 times. Instead of that, we just have these things where we put the genes in so that the corn or the soybeans or the canola is able... Wait, not the canola. That's herbicide tolerance. The insect resistance, the cotton. There's a BT cotton that's really helped in India quite a bit in herbicide tolerance. But where was I going with that? Yeah, we can eat this stuff just fine because we're not insects. People are saying, oh, it's dangerous. Glyphosate is dangerous. That's a separate story. The BT is dangerous, and it's not to us. But glyphosate is not. It's not dangerous to us. And so the very trace amounts that are below minimal effective dosage or whatever, way below actual limits are. And there's some question about what kits they're using and how they're misapplying that. But that's, again, another story. The papaya, You could argue it, but it looks like it was pretty much flat out saved in Hawaii because of the genetic modification to confer disease resistance. I think it was to a fungal pathogen. Because we have a bunch of crap we use soybeans for. And so sugar beets. This is going to be, this is going to be Roundup Ready of some variety. And of course, sweet corn, we have cultivars of those that have those traits. BT and glyphosate resistant, and summer squash. Unless this has changed in the last year, these are the only GMO things that we have out there. But I am going to mention, in the organic food world, there's a method that is approved and allowed for organic farming, and that's random mutagenesis. You can take and you can expose your seeds or your seedlings to gamma radiation is one way to do it. You know, induce mutations. Remember looking at the DNA repair mechanisms there. Or chemical mutagens. You can expose your seeds to that and grow them up. And if the mutations have conferred a new trait that makes it more marketable, you can still call it organic. Random mutagenesis by radiation, still organic. All right, so Agrobacterium tumefaciens is still the the shuttle vectors, at least. You don't have to use the Agrobacterium to confer those. And the organic insecticide produced by Bacillus thuringiensis, that's the BT toxin. Now, there was a tomato that was approved, and that was from 1994 is when it was approved. It's called a flavor saver tomato. And it failed because Attack of the Killer Tomatoes, right? It's a GMO, so it must be dangerous or harmful in some way. Aaargh! God, that was terrible. I used to be a better actor than this. I swear. At least I was told that. But then again, I was in my 20s. Anyway, no. So it failed in the marketplace. There were problems maintaining it and problems with shipping. So it failed in the marketplace. Now, one thing that I wish that we had available right now that Canadians are enjoying by the ton is the AquAdvantage Salmon. So this is a genetically modified salmon that is able to be farmed and grow to adult. Here we go. Or right here, the farm salmon, both at 18 months old. The farm salmon's about halfway grown up. The genetically modified salmon is a young adult right there now. Done. Boom. And now how this is accomplished, this AquAdvantage Salmon, I don't know if I like that portmanteau, to be honest, because it's aqua, and that's fine water, and then advantage, so AquAdvantage, AquAdvantage, I don't know, this is messing with me a little bit. Anyway, so we made this, they took this, they made a DNA construct of the growth hormone gene from the Chinook salmon, and put a promoter from the ocean pout. So you have a promoter from the ocean pout in front of the growth hormone gene from the Chinook, and that was put into the Atlantic salmon. So again, 18 months rather than 3 years to adult side. Now the facility is in Panama and it's landlocked, so it's not like these salmon are getting out into the environment at all. Even if they did, well, no, they would outcompete the crap out of native salmon. So never mind. We need to keep them landlocked and out of the way. But yeah, I would love to try this sometime. Oh, wait, maybe I can. I think July, we're planning on going to Washington State, and I'm trying to get my passport renewed and get passports for the boys. But we might take the ferry over into Victoria, say, and maybe somewhere up there they're selling the AquAdvantage salmon. So maybe I can try this. And then maybe I can post about it. And if for some reason you're still watching any videos I make, I'll share that. Or I'll share it on, I don't know, whatever. Anyway, I have a chance to maybe try that. That'd be fun. All right, now remember those apples. So when you eliminate this polyphenol oxidase gene, now this says reduced. Maybe it is only reduced. Maybe they mess with its ability. Oh, that's what they're indicating here. So it's it can't be transcribed as frequently as how I imagine that you would do that. You mess with its upstream activating sequence or some other promoter aspect, and then perhaps that's how you get a much reduced the polyphenol oxidase gene. Okay, now lab-grown meat. This goes back to 2013. This is in the petri dish. Allegedly, this is okay. Now it's supposed to look like beef because it's supposed to be it's striated muscle tissue, right? That's grown. A clean. Wait, what's the phrase they're trying to market? There's this company called Memphis Meats that's probably the leader in all this. And they're trying to go with clean meat instead of vat meat or lab-grown meat, or lab meat. Because obviously branding matters, right? And I mean, it does. If you say lab-grown meat, people are going to avoid it. Because that's people are people. And why does it seem we should get along so awfully? Because we're people. But anyway, that's a Depeche Mode reference. Oh God, I'm sorry that I'm umming. I hate that when people speak. They have this cost down now. So when they first made this cultured hamburger, the cost was estimated at about $300,000. They've got it down now to it's $1,200 per pound, which is still, oh wait, it was a $2,400. It was like $600 per quarter pounder. So it's still not commercially viable yet, but we're getting there. And so that should hopefully remove a number of the ethics concerns of slaughtering animals to consume their flesh. The issues, the other issues around this have been the finding the right kind of additives, because without fat at all, apparently it tastes terrible. And so they're coming up with other options for that, to have some kind of to help that meat taste. But anyway, that's not quite there yet, but there's another potential application for this kind of technology. All right, so the Impossible Burger, there is a version 2.0 out now, and as you can see here, it looks very burger-like. This is accomplished, so this is a mixture of a number of things, it's vegetarian, it's completely vegetarian, and that's probably why they used this. So they took a yeast, they modified it to produce leg hemoglobin, which is a type of heme molecule that is produced by soybeans. So it's a soybean heme. And that's what gives it the meaty. That's what gives meat the meaty flavor. That, in fact, when you're squeezing out that steak, why would you squeeze out the steak? That's a terrible example. The burger, whenever you see that red mess left behind, that's not blood so much as heme and whatnot. So anyway, the Impossible Hamburger. I've had this, the Impossible Burger. I had it a year ago at Morel's here in Louisville. It was like a year and 2 days ago now. I don't remember that because of Facebook memories, not because I remember that. That would be kind of psychotic. Well, okay, not psychotic, but it'd be weird. Anyway, I tried the version 1, and I'm looking forward to trying version 2.0 that's been released. So hopefully I get to try that in the next little while. But it is tasty. I definitely recommend trying it out. Okay, whole genome sequencing, as I mentioned before. Now, at first we did this because we had to build these libraries using bacteria or even bacteriophages, and then we had to Sanger sequence all the distinct individual inserts from that library, and then we had to assemble it, stitch it together based on overlapping sequences. Very tedious, very long, took forever. So we only did that for certain types of organisms, and we stuck with the model organisms that we use for so much other stuff. So some Arabidopsis for plants, Saccharomyces for our yeast, of course the mouse, which and then Drosophila, and C. elegans, Caenorhabditis elegans, which Dr. T works with. Oops. So that's why we have these, well, that's not why we have model organisms. That's a whole nother, having a common organism to share research efforts around to figure things out that are common to all of humanity. Because as Monod said, what's true for the fly is true for the elephant. I think it was fly. Because I'm looking at the fruit fly. Okay. Yeah. Oh, hey, we're almost out of time. So How do we apply genomics? So predicting disease at the individual level sounds great, but what about privacy? What about insurance? Do they have a right to that data? We also have a bunch of genome-wide association studies where we use single nucleotide polymorphism. So these are individual nucleotide differences in areas of genes that don't vary very much, that don't change rapidly. And we can use that to map populations and other things too. International HapMap project being one. And pharmacogenomics, the idea that we can tailor medicines to the individual, which sounds like something a really rich society would have. But then again, I'm in America where healthcare is apparently more of a privilege than a right. Maybe in someone teaching this class in Finland is like, oh yeah, pharmacogenomics will be great because then we'll be able to help people more, and they'll be able to help everybody. They'll be able to believe they can help everybody. Anyway, I want to believe that someday myself, right? So you can use these SNPs to identify ancestry, and I have done this with the 23andMe service. This is for entertainment use only, realistically, because there's no, what's the value in this data and this information? One of the reasons I joined 23andMe and I did this was because my biological father had a number of wives and a number of offspring, and I'm just expecting someday to come up with a half sibling out of nowhere, or other half, other relatives from other affairs he had. Apparently, he could convince a lot of women to have relations with him, and they were the kind of women, and he was the kind of guy that didn't much think about birth control, apparently. Or maybe he did, and these are just, that's a whole different spin on that scenario. It's not worth going into. Anyway, the other thing I think is fun. Now, this is for fun. There are some people that were trying to use this to prove how white they were. I don't know. I didn't need evidence personally, but I'm not one of those groups that holds that confers anything special, intrinsically special. Institutionally, lots of special, right? Which is wrong. Which is one of the problems, or the major problem in our society. Anyway, the other aspect of this that's fun are the Neanderthal variants. And so, because we didn't just kill the Neanderthals, other stuff, right? We interbred, especially the Europeans. Populations from Asia and India too, I believe, are mixed with another early hominid, the Denisovians. And yeah. So metagenomics, again, we're taking, this is how we can identify a bunch of species in a population. We can just do it by taking DNA sample from soil, for example, and you can get a representation of what species are in that soil and to what, how populous they are in that soil. That's not perfect. So biofuels, again, renewable fuels, the Navy is looking at this too. And that's where we're getting some of our current drivers for climate change, is the fact that it's a legitimate national security issue as well. So biofuels is something definitely of interest to the military, and it should be of interest to more people, but here we are. But speaking of power, nuclear power works great. We've been doing it for 60 years now. And, we've been doing it for so long, and it's been done so widespread across the world that when you think of nuclear accidents, you can only think of four or five? Wait, Fukushima being the most recent, and we have 3 Mile Island, and we had Chernobyl. We had SL1, which you might not know about. It's not classified anymore, but it was a test reactor we gave the Army in the desert of Idaho that they blew up. They promptly blew it up. It was small, but yeah, that's a nuclear accident. And where else? We lost a few subs, a couple of subs to other things that weren't. Anyway, nuclear power should be part of our strategy for long-term energy that's not oil dependent. I'd be happy to just talk about that in class sometime. Maybe Wednesday. Maybe we'll talk about some other stuff. Maybe we'll talk, make it a conversation. Anyway, because you have the midterm coming up on Friday, and then everyone else that wants to do these other questions, these take-home questions, you'll be doing that before then. But anyway, So your mitochondrial genome, you know, that's matrilinear. Now I can tell. I didn't tell this in the 101 class, and I'm not going to, but there are some instances of paternal mitochondrial inheritance in humans. It's kind of neat. Look it up. Forensic analysis, so Bacillus anthracis. We tracked down the guy that was growing this all up in his lab, his basement lab. I still can't believe I said tuberculosis when I first told you about that. I'm like, that's so dumb. TB is a terrible choice as a vector for a biological weapon. It takes way too long. Not that I spent a lot of time thinking about that. So again, transgenic plants. These are plums, resistant to the plum pox virus. I don't know if these are on the market, though. And then proteomics, of course, where we look at the proteins. And I don't know a ton about proteomics, I'll be honest. But the idea being that we could take very low levels of analyte, like even on your breath, if you have like tumors in your respiratory system in any way, where you can take tiny small amounts of analytes and you can give a good strong predictive yes or no on tumor or not tumor. The efforts as of the writing of this text indicated that that's not the case so much, that there's a lot of false positives and whatnot, but this is still a work in progress. And that's all I've got. I will find a good way to end these someday. Like if I mouse over here while I'm talking. I will see you on Wednesday. And I should have something for you if you want to take home midterm by this afternoon. It'll probably be after lunch though, because it's already, yeah. All right. Anyway, have fun. And again, you can email that too, but you can use a discussion group for any questions too, if we wanted to interact like that. All right. Enough of that. Bye.